By the name of the God, let's start our lecture today. Our lecture today is about small intestine. By the end of this lecture, we're going to be able to know the different parts of the small intestine, which are duodenum, jejunum, ileum. You will also be able to identify the cell type lining of the all intestinal part, including villi and crypt. Let's start our lecture today with the small intestine. Small intestine is the site where we complete digestion, complete nutrient absorption, and endocrine secretion. The length of the small intestine is about 6 meters long. This 6 meters long is going to be divided into 5 parts. First part will be the duodenum, which will be the first part in continuation with our stomach. It will be about 25 centimeters long. It takes the C-shaped structure. In the lumen of this C-shaped structure, you will find the head of the pancreas. The second 2/5 will be the jejunum, which is about 2.5 meters long. The third part and the remaining part will be the ileum, and it will be about 3.5 meters long. Now let's talk about our small intestine. Our small intestine is a tube covered by peritoneum. It is a gastrointestinal tube covered by peritoneum. When we say that it is covered by peritoneum, it means that the fourth layer or the outer layer of the tube will be serosa. So what are the 4 layers of any gastrointestinal tract tube? They are going to be mucosa, submucosa, muscularis externa, and finally we have serosa. When we are going to look to the inside of the tube to look at the general structure of the T, we are going to find that the mucosa or the inner side is not smooth. It having folds. This fold is giving it velvet-like appearance. It is not clear. When we are going to see velvet-like appearance or this circular structure, the circular structure is called plicae circularis. So what are this appearance? It is a plicae circularis. Okay. When we are going to have a model for this plicae circularis, we will find that it is a fold in mucosa and submucosa. We are going to have this circular fold. What is it formed of? It is formed of mucosa and the underlying submucosa forming fold. This fold is a circular fold along the whole diameter of the tube. This is called plicae circularis. Moreover than that, when we are going to see this fold, when we are going to look at the mucosa, we are going to make the line of the surface. This is the line of the surface. Above this line, there is a finger-like projection. This finger-like projection is called villi. And below the surface, there is a deep invagination, deep downgrowth of the epithelium. This is called crypt. So, as regarding the mucosa and submucosa, mucosa and submucosa are forming a fold, a circular fold, making the appearance of the inner surface of the tube velvet-like appearance. The circular fold is called plicae circularis, formed of mucosa and submucosa. When you are going to looking at the mucosa, mucosa will be formed of the 3 layers: epithelium, lamina propria, and muscularis mucosa. We will find that the epithelium and the lamina propria are going to make a fold, making finger-like projection above the surface. This finger-like projection will be called villi. Not only that, but also they will make a downgrowth, making a deep invagination below the surface, and this is called the crypt. Can you guess why? Why we are making circular fold? Why you are making villi? Why we are making a crypt? In order to increase the surface area. For what? For absorption. As we mentioned before, one of the functions of the small intestine is completing digestion and completing absorption. So we need a huge surface area for doing this process. How we accommodate this, how God help the intestine to do this function? The small intestine is a long structure. It is about 6 meters long. Not only that, but also it is making a fold. This fold is circular fold called the plicae circularis. Not only that, but also the mucosa is making finger-like projection called villi and deep invagination called crypt. Let's go for more naked eye appearance of this plicae circularis and villi and crypt. Now let's see a live naked eye appearance when we open the small intestine. When we open the small intestine, we find that there is a fold. This fold is called plicae circularis. This is maybe similar to the Roget of the stomach. Do you remember Roget of the stomach? But Roget was just a fold, and it can be lost due to extension of the stomach. But this plicae circularis is a permanent feature. When you open the small intestine anytime, you will find this plicae circularis if anywhere, and you will find it more in the duodenum and in of the proximal half of the ileum, duodenum, jejunum, and the proximal part of the ileum. You are going to see it well developed, and it's going to be less in the distal ileum. Okay, and when we are going to see it by histological structure, we are going to see that there is upward growth or finger-like projection, which is formed of core of some mucosa and mucosa. This is called villi. Downward growth below the surface. This is called the crypt. Does crypt and villi and the crypt disappear? No, they are also permanent structure. So any structure in our small intestine will be permanent, starting from plicae circularis, villi, and crypt. Let's go more for more higher magnification for this villi and crypt. Magnification for our small intestine. Our small intestine is formed of, as a tube, is formed of mucosa, is formed of some mucosa, muscularis externa, and finally we have serosa. Do you remember what is the characteristic feature of the small intestine? Yes, it is a fold of mucosa and submucosa. This fold is forming plicae circularis, and there is also elevation in the submucosa above the surface. This is called villi. There is also a depression below the surface or invagination, and this is called crypt. Now let's talk about the general structure of a small intestine. As a small intestine, it is a gastrointestinal tube formed of the four layers. The innermost is the mucosa. As we mentioned in the previous slides, we say that the mucosa is making a finger-like projection above the surface. This finger-like projection was called villi, X-line, and there was a down growth under the surface, and which was invagination, and it was crypt. The scientists which find it named it cleft of Riper cone. So mucosa is formed of villi and crypt. We can consider this crypt as if it is a simple tubular gland. Okay, what about the mucosa? How formed of how many layers? Do you remember? Yes, it is formed of 3 layers. What are they? They are the epithelial lining, the connective tissue lying beneath the epithelium, which was called lamina propria, and finally we have the muscle which moves the mucosa, and it is called muscularis mucosa. Our epithelial lining here will be for the function of absorption and secretion, and it will be simple columnar epithelium. As regarding the lamina propria, here the lamina propria is a loose connective tissue extending into the core of the villi and filling the spaces between the crypt. It is very rich in blood capillary. Do you know why it is rich in blood capillary? It is an important piece of information because small intestine is going to absorb food. So for this absorbed food, it will be must be go to the blood capillary. So here lamina propria will be very rich in blood capillary. This connective tissue corneum is very rich also in lymphocyte, plasma cell, and macrophage. Is this logic? Yes, because this is how we are eating food. Maybe not cooked well, maybe containing virus or bacteria. So we need an immune cell to defend against your body. So that means that the lamina propria here is very rich in blood capillary, containing many lymphocyte, plasma cell, and macrophage. Finally, we have the third layer, which is the muscularis mucosa. It is smooth muscles which are arranged as few layers of inner circular and outer longitudinal muscle. Now let's talk about the second layers of the tube. The second layer of the tube is the submucosa. As we mentioned before, submucosa is a dense connective tissue containing blood vessel lymphatics and also containing mesenteric nerve plexus, which is called Meissner plexus. Here we have to stop on the submucosa because it is an important layer in the small intestine. Why? Because we have in the small intestine three parts. They are duodenum, jejunum, and ileum, as we mentioned before. How can we differentiate histologically between these three parts? All of them is formed of mucosa, submucosa, muscularis externa, and serosa, both of them having the mucosa containing villi and crypt. So, to differentiate between them, we have to look at the layer which is called submucosa. This submucosa will have a specific structure for each part of the duodenum, of the part of the small intestine. For example, in the duodenum, if you look at the submucosa and see that it has a mucus acini, this mucus acini is called the Brunner's gland. So this will be the duodenum. If we look at it and find the submucosa is just connective tissue containing nothing, so it will be the jejunum. If you look at submucosa and find that there is a lymphatic aggregation, lymphatic follicles, this lymphatic follicle is called Peyer's patch. So this is ileum. So how to differentiate between duodenum, jejunum, and ileum? Yes, we look to the submucosa. Submucosa in the duodenum you will find mucus acini, which is called Brunner's gland. So this is duodenum. In jejunum, submucosa will be empty, containing just connective tissue, nothing specified. What about the ileum? I will find lymphatic follicles called Peyer's patch. Okay, can you see these two photos? These two photos, in the submucosa here, I find rounded structure, rounded pale structure. This is mucus acini, which is called Brunner's gland. So this is duodenum. Type. Look to the second photo. This one. Here it is making a circle around lymphatic follicles. This lymphatic follicles is called Peyer's patch. So this is ileum. So this is a photo of the duodenum and this is a photo of ileum. What about musculosa or muscularis externa? It is the main layer, main muscle layers, which is formed of inner circular and outer longitudinal, embedded in a connective tissue containing a mesenteric nerve plexus, which is called Erbach plexus. Here, the function of muscle is very evident. We are here calling it peristalsis. This Erbach plexus, which is embedded in between the inner circular and the outer longitudinal, will control this peristaltic movement. Finally, we have this last layer, which is the serosa, which is a connective tissue, in addition to the simple squamous epithelium, which is called mesothelium, because small intestine is covered by peritoneum. Here we will start to talk about the intestinal villi. As we mentioned before, it is a finger-like projection above the surface. It is formed of a core of the connective tissue called corium. So here we are going to have a core of connective tissue. This core is called corium, covered by epithelium from outside. It differs in shape according to the site. If you are in the duodenum, it has a shape. If in the jejunum, another shape, and the ileum, there is a different shape for the villi. That's to say, for example, we mentioned before the submucosa of the ileum is having a lymphatic aggregation called Peyer's patch. Villi is absent over this Peyer's patch. So, finger-like projection above the surface is called villus. It is formed of a corium of connective tissue covered by epithelium. What about the corium or the connective tissue of the intestinal villi? It's considered as loose areolar connective tissue extending from the lamina propria. It contains, as we mentioned before, extensive capillary, lymphocyte, plasma, and macrophage for the immune cell. Also, it has few smooth muscle fiber. Okay, so connective tissue of the corium is loose areolar connective tissue containing blood capillaries, many blood capillary, lymphocyte, plasma cell, and macrophage, and don't forget smooth muscle fiber. The third component of the villus is the central lacteal duct. What is central lacteal? If we see here, this is a scanning electron microscopic picture or 3D figure for the villi. As we see here, it is a corium of connective tissue. Here we have a dilated duct. This dilated duct is important for absorption of fat because fat is large molecule can be absorbed into the fenestrated blood capillary. So we have a dilated duct in the center of the corium or of the villus. This is called central lacteal, around which we can find this structure. Can you guess what is this? This is the smooth muscle fiber we mentioned in the previous slide. Why? As we absorb fat, it will be moved into the central lacteal duct. In order to move fat into the circulation, it must be aided by the contraction of the smooth muscle fiber in the corium. Other components such as protein, such as carbohydrate, will move to the circulation through fenestrated blood capillary. These capillaries is numerous and forming extensive network. This is fenestrated. Why it is fenestrated? It means that there is a pore in the endothelium of the blood capillary. Why it is fenestrated? In order to allow absorption of food into the circulation, to make channel for passage of large molecules such as protein, such as carbohydrate. Was the structure of the corium, which was a loose connective tissue containing cells of the immune system, containing smooth muscle fiber, central lacteal duct, and fenestrated blood capillary. Now talk about the coverage, the epithelial coverage of this villus. The cell lining, the epithelial coverage are going to be of three types. What are they? The first one is the absorptive cell, which is responsible for the absorption of nutrition. This cell, as it is in the small intestine, it will be called enterocyte. We are going to have another cell which will produce mucus for lubrication. It will be goblet cell. And the cells which secrete hormone into the circulation and distributed among the enterocyte is called enteroendocrine cells. So what are the cell lining the villus? There are three types. What are they? Absorptive columnar cell, goblet cell, and enteroendocrine cells. What about crypt? What are the cell lining the crypt? As we mentioned before, crypt, as we mentioned before, is deep invagination of the epithelium into the lamina propria. This deep invagination is called a crypt. This crypt is considered as a simple tubular gland, occupying the thickness of the lamina propria of the connective tissue. It is present among in between the villi covering the epithelium. Okay. The epithelium lining the intestinal crypt. There are about 6 types of cell lining the crypt. 3 are continuation of the intestinal villus. Do you remember the 3 cells covering the villus? They are columnar absorptive cell, enterocyte, they are goblet cell, and enteroendocrine cell. Here we have a different 3 other cells. What are they? One of them is Paneth cell. We are going to have stem cell, undifferentiated stem cell. We are going to have a cell called M cell, membrane-like epithelial cell. So crypt is lined by 6 different types of cells. Okay, let's talk about each cell separately. Now let's talk about the first type of cell lining the villus and the crypt, and it is the commonest. It is columnar absorptive cell, or the other name it is enterocyte. Where can we find this cell, in villus or crypt? In both villus and crypt. But in the crypt, you must find it in the upper part only. So in the upper part of the crypt, you will find columnar absorptive cell. What about the shape of the cells? Shape of the cell, all the epithelial lining of the smallest intestine is simple columnar epithelium. So this cell is going to be columnar cells with oval basal nucleus. When we are going to stain these cells by hematoxylin and leucine, it will be acidophilic. But acidophilia will be more on the upper surface. It appears as if it is like that of the brush. So it has apical acidophilic brush border, or another name, it is striated border. This apical acidophilic brush border is having a thick glycocalyx coat. A glycocalyx, we mentioned the word glyco. So it will be BAS positive stain. Okay, here, if we are looking here to a villus, this is the corium of the crypt. It is covered by all the cell is simple columnar epithelium. The most common one is a columnar absorptive cell. It appear acidophilic. Can you see here the apical part of this enterocyte? It appears as deep acidophilic. It is a brush border. Why it is a brush border? This must know the electron microscopic photo of the enterocyte or columnar absorptive cell. Now let's look at the electron microscopic structure of the enterocyte or columnar absorptive cell. In order to talk about it, we are going to see that the shape of the columnar absorptive cell appear as columnar cell, as we mentioned before. It has active nucleus. What about the organelle? We will find well-developed smooth endoplasmic reticulum. We will find mitochondria. We will find a network of RER and Golgi apparatus. We can see here few dots of free ribosome type. Look at the top of the cells where we mentioned that it is deep acidophilic appearing striated. Why it appears striated? Because when we look at the top surface, what can you see? We can see here numerous microvilli, about 3,000 microvilli. This is apical microvilli in order to increase the surface area of absorption. It is a finger-like projection from plasma membrane. Can you see here? Moreover than that, what is this? This is thick cell coat, thick glycocalyx. This is why it appears BAS positive, because we're having a glycocalyx. Can you see here? Numerous microvilli with a thick fuzzy appearance of glycocalyx or cell coat. Here, this is the microvilli marked in red, finger-like projection covered from outside by a cell coat. Let's go to see more about. We are having more zooming on the microvilli. It is finger-like projection on the surface of the cell. It is covered from outside by a thick glycocalyx coat. Is it important? Let's go to talk about the structure of the microvilli and what is it. Now let's talk about the structure of microvilli. Microvilli, as we mentioned before, is a finger-like projection from the surface to upper surface of the cell. It is about 1 micrometer long and about 1 micrometer in diameter. طيب, what about the core of microvilli? What make it keep its shape? It is a permanent structure. So what make it a permanent structure, preventing it from becoming a flat surface? What make it is the presence of a core inside the microvilli. Can you see this core? This core is formed of actin filament. This actin filament is not only the core, it also anchoring with another actin and intermediate filament, making a network beneath it. This network is called terminal web. So, microvillus contain a core of actin filament. This actin filament of the core is anchored with another actin filament and intermediate filament, forming a network which is called terminal web. It is present beneath the microvilli. We mentioned before that this microvilli is covered by a thick coverage of glycocalyx or cell coat. What is the importance of this glycocalyx or cell coat? This cell coat, one of the most important is that it protects the microvilli, protect it from autodigestion. Here you are in the small intestine, we will receive hydrochloric acid from the stomach, we will receive pancreatic enzyme. So this can digest our microvilli or our cell. It is protected from autodigestion by the thick cell coat. Also, we are going to have enzyme in this cell coat. This cell coat will form enzyme which is called brush border enzyme. For example, it is dipeptidase and disaccharidase. You know from biochemistry that this enzyme will make complete digestion of the protein coming from the small intestine and carbohydrate and help in terminal digestion. We mentioned before is one of the functions of the small intestine is making terminal digestion. For example, dipeptidase will digest protein, remaining protein. Disaccharidase will digest carbohydrate into monosaccharide for easy absorption. So What is the function of glycocalyx here? It is an important question. Microvilli and glycocalyx. Glycocalyx protect the microvilli from autodigestion, also the brush border enzyme, for example, dipeptidase and disaccharidase. Okay. Now let's go about the borders of this columnar cell or this enterocyte. We will find that this lateral border is going into interdigitation in order to allow the cell to be firmly attached to each other. Not only that, but there is a junction complex. Do you remember junction complex? It is formed of three types of junction. They are zonula occludens, zonula adherens, and desmosome. It is ZOZA D. Okay, so in the lateral side, we are going to find lateral interdigitation. More than not only that lateral interdigitation, we are going to have a junction complex. Don't miss it. It is zonula occludens, zonula adherens, desmosome or macula adherens. Why we are going to have junction complex, especially zonula occludens here? Because we are going to allow passage only of food, absorbed food through the cell. It is not allowed to pass in between the cells. So this will prevent the passage of the absorbed food through the intercellular spaces in between the cell. It is only to allow it to pass through the enterocyte or the simple columnar cell. We mentioned before that small intestine, one of its functions is completion of digestion. Completion of digestion of protein and carbohydrate, and this was completed by the presence of brush border enzyme in the glycocalyx. For example, we have dipeptidase and disaccharidase. After completion of digestion, this amino acid and glucose molecule will go into the circulation through the fenestrated capillary network present in the column of the villus. What about absorption of fat? Fat will be absorbed in the form of chylomicron, and from biochemistry, you know that chylomicron is a very large molecule. How we reach this chylomicron? We are in the enterocyte containing many organelles. We mentioned before that we have SER, we have RER, and we have Golgi. How to deal with this fat? Which organelle do you know that it deals with fat? Yes, it is a smooth endoplasmic reticulum. Here we are going to have smooth endoplasmic reticulum which will deal with fat and resynthesize triglyceride. After that, we are need to carry this fat over a protein. What organelle can involving this protein? Protein is synthesized by rough endoplasmic reticulum and then go to the Golgi apparatus in order to be incorporated with or combined with this triglyceride. Combination of triglyceride with protein will form a molecule called chylomicron. Chylomicron is a large molecule, can't be passed directly into the fenestrated blood capillary because it is larger than fenestry. So we need to indirect pass this chylomicron into the circulation. How? This chylomicron is going to move through the basolateral border to where? To go into the corium in the connective tissue core of the villus, where they will find a large duct which is called central lacteal duct. Do you remember this? It was in a previous slides. In the center of the corium of the core, there is a central large duct. It is called central lacteal duct, where chylomicron will pass through this central lacteal duct to go to the thoracic duct and pass indirectly into the blood stream. Okay, here we have complete talking about absorption of different food stuff, including carbohydrate, protein, and finally fat. Fat need incorporation of many organelles. It needs cooperation of smooth endoplasmic reticulum in order to resynthesize triglyceride. RER will synthesize protein, which will be conjugated in the Golgi apparatus with this fat to make large molecule called chylomicron, which moves through the basolateral border into a large duct in the corium, which is called central lacteal duct. After central lacteal duct, it will move to thoracic lymphatic duct, and finally reach to the bloodstream indirectly. Now, if we are going to have a disorder of the small intestinal mucosa, what will happen if we are going to damage this mucosa? If we damage the mucosa, if we damage the villi, so there is a decrease in the surface area of the stomach. So we will not be able to make complete absorption of our food. So this is considered one of the disease which of malabsorption syndrome. We have a problem in absorption, so it is a malabsorption syndrome. One of the diseases of the malabsorption syndrome is celiac disease, which may cause damage to the villus, resulting in malabsorption. This is caused by immune reaction. It is an abnormal immune reaction about eating gluten. When they eating bread or one of the flour component, they are going to have immune response against this gluten. So they will cause damage to the villus. This immune response will make damage to our villus. This will may cause malabsorption syndrome. We will not able to absorb our food. This will cause unabsorbed food will make diarrhea, abdominal pain, and red growth because we can't absorb all what we need from food. What about gluten? Do you know gluten? It is found in bread, in any component formed from wheat. Okay, so here we are going to have a clinical application called malabsorption syndrome. Malabsorption syndrome is a large category of disease. One of them is a disease called celiac disease. The celiac disease results from abnormal immune reaction against gluten. This will lead to abnormal immune reaction which will cause damage to our villus, resulting in decreased the surface area of absorption, resulting in malabsorption syndrome. Now we are going to talk about the second type of cell which is present in on our villus. It is like the simple columnar epithelium. It is present in both the villus and crypt. Also, here it is not present all over the crypt. It is present only in the upper part. This cell is called the goblet cell. Do you know goblet cell? We take it in the respiratory system. This cell which produce mucus. Can you guess what is the function of mucus here? It is mainly for lubrication. So this cell will be found among the columnar cell all over the villus and in the upper part of the villus. It is considered as gland of one cell. It is a unicellular gland. What does this cell produce? It produces mucus. It produces mucinogen. It is a glycoprotein. Can you guess? As we have a glycoprotein, what are the organelle we need? It is as we need protein, we need ER. What else? Golgi, mitochondria. This organelle will produce mucinogen for us. Okay, this mucinogen will be absorbed, will be stored in the upper part of the cell. So where to find mucinogen or mucus granules? It will be apical in the cells. What about the organelle? They are going to be basal. Excellent. We mentioned before that all the epithelial lining of the small intestine is simple columnar epithelium. What about the cell? If you are going to have simple columnar cell making storage of mucus, this mucus will cause it to be having expanded upper part. Are you seeing here? This is a columnar cells. This is a columnar cell having storage of mucinogen into the upper part, causing the upper part to be expanded while the lower one is narrower. So this having expanded apical part because of the presence of stored mucinogen granules. What about the basal part? It is narrower, so it is like goblet, so it is called goblet cells. What about cytoplasm? Cytoplasm will be apparent in the lower part. Is it acidophilic or basophilic? It is basophilic cytoplasm. Why it is basophilic cytoplasm? Due to the presence of rough endoplasmic reticulum. What about nucleus? Are you going to find it apical or basal? No, it will be present because the apical part contain only mucinogen granules. Okay, what about staining by hematoxylin and ecine? You are going to, it is simple columnar epithelium with expanded apical part. Can you see the apical part here? It appear vacuolated. Why it is vacuolated? Because during the preparation of hematoxylin and ecine staining slide, you are going to see that there is dissolved mucinogen. This dissolved mucinogen appear in the slide as if it hasn't been vacuolated. So what about electron photo of this cell? What to find in the apical part? In the apical part, you are going to find electron-lucent granules of mucinogen. Excellent. Here you are going to have electron-lucent granules. What about the basal cylindrical part? We are going to find achromatic nucleus. We are going to find abundant RER with Golgi complex. We are going to find mitochondria. What is the function of this cell? It is going to secrete a glycoprotein. What is the other name of glycoprotein? It is mucin. What is the function of mucin? It is for protection and lubrication of the wall of the small intestine. Why is it important? Because to avoid injury to the wall. And you are going to find that as we are going downwards through the intestine, we are absorbing water from the food. So food become hard. So we need more lubrication. So as we are going down, you are going to find there is increase in number of goblet cells. So in the duodenum, you will find the smallest number of goblet cells, while in the jejunum and the ileum, there is more increase in the number of goblet cells. By this, we have finished to the end of the goblet cell. Now we are going to move to another cell, a Pannis cell. Pannis cell, if you remember, it is present in crypt only. It is not present in villus. Okay, so where to find Pannis cell? Pannis cell is one of the cells present in crypt only. It is present where in the crypt? In the upper part, as we mentioned before, we have simple columnar enterocyte and the other cell which is goblet cell. What about the base? The base occupied by a cell called Pannis cell. This Pannis cell is a typical protein secreting cells. It is a pyramidal or high columnar cell having basal rounded active nucleus. What about cytoplasm? As I am saying that it is typical protein secreting cells. Typical protein secreting means that we're having the organelle of protein secretion, which is, yes, RER, Golgi, mitochondria. This will make the cytoplasm basophilic. And we are going to store this protein so it will appear as apical acidophilic granules. It is apical acidophilic zymogen granules. See this, what we say is right or wrong. We say that this is Pannis cell. Pannis cell is a typical protein secreting cells. This is what we say. What can we see by electron microscopic? You are going to see that there is apical granules, apical electron dense granules. This granules is the stored protein which will be discharged to the lumen by exocytosis. What about the organelle? The organelle present is RER, as you are going to see here in this photo. You are going to see RER, Golgi, and mitochondria with achromatic nucleus. What is this granules or what is this protein which is going to be discharged out of the cell by exocytosis? To say what is this protein function of this cell? This cell secrete lysozymes. Take care here. This is lysozymes, not lysosomes. Lysozymes is an antibacterial peptide. Lysozyme is antibacterial peptide. Okay, not lysosome. Lysosome is written like that. This is a peptide called lysozyme. It is an antibacterial. In addition to lysozymes, it is an antibacterial peptide or cryptidine. It regulates bacterial flora of the intestine. Normally, we have bacterial flora in our intestine. What prevents it from being pathogenic or overgrowth of this bacteria? It is the peptide secreted by the Pannis cell, which is lysozymes. It is an antibacterial peptide. What about other cells we mentioned in the villus and crypt? we mentioned that there is secrete hormone which is enteroendocrine cell. We mentioned this cell before in the stomach. It is, as we mentioned, it is present in the villus and crypt, but it is more numerous in the base of the crypt. It is all over the villus and the crypt, but it is fewer. Or so where to find it more or more numerous? It will be in the base of the crypt with Pannis cell. As we described previously, we described it with the fundic gland. It is characterized by being a cell which will secrete hormone. Secreting it where? Into the lumen of the intestine? No, it will be secreted into the blood capillary present in corium of lamina propria. So this granule is characterized by having a granule where apical or basal. Basal, why? In order to be secreted into blood capillary. So it is characterized by an important feature. What is it? It is basal secretory granules. They will secrete variety of gastrointestinal peptide hormone which affect other cells. Can you see an electron microscopic photo for this enteroendocrine cells? Now let's see electron microscopic photo. What can you see here? You can see here basement membrane, basal part. We can see here simple epithelium, simple columnar epithelium. We are seeing some nucleus at the same level with the apical part of the cell showing microvilli. Can you see here this cell? This one? What can you see? We are seeing here basal electron dense granule which are going to be poured into blood capillary. So this is an endocrine cell. This is an enteroendocrine cell. What is characteristic for enteroendocrine cell? The presence of basal electron dense granule. What are the hormones can be secreted by this enteroendocrine cell? It can produce various hormones. What are the hormones that can be secreted by this enteroendocrine cell? It can secrete various of enteroendocrine hormone. One of them is EC cells, which secrete serotonin. It can secrete by I cells. It is a different type of enteroendocrine cell according to the secreted hormone. D cell can secrete somatostatin. We have G cells we mentioned before, which secrete gastrin. We have K cells, MO cells, which secrete motilin. N cell, which secrete neurotensin. S cell secrete secretin. L secrete glucagon lipopeptide, and so on. It is a variety of hormone and having different action on neighboring cells. Now let's talk about another type of cells, which is stem cell. It is responsible for regeneration and it is a stem cell. It is undifferentiated stem cell. It is present in the crypt base. So can you guess what are the more abundant cells in the base of the crypt? Yes, one of them is Pannis cell. Excellent. Enteroendocrine cell, and the third one is the stem cell. It is found mainly at the base of the crypt. It will go under many divisions which migrate to regenerate different type of cells of the villus and the crypt. It will be found in the base and go upward to migrate upward in order to make regeneration for all types of cell. It can make complete regeneration of the cell of the villus and the crypt in nearly 5 to 7 days. As we know about any stem cells, as is having a basophilic cytoplasm, vesicular active nucleus with prominent nuclei. It is considered, as we are speaking in the small intestine, all of them are columnar, but here as it is a stem cell, it will be SS, it will be short columnar cells. What about the organelles present? The more important organelle present is the free ribosomes, which may be in aggregation so-called polyribosome. So don't miss many ribosomes. It will not be attached by many junction to the outside. It has a smooth lateral border, no lateral interdigitation with the cell in order to allow migration and division into other types of cell. So what is the function of this cell? The main function, as it is a stem cell, it is regeneration of all lining of cells of crypt, as it is in the crypt, no, of both crypt and villi. In order to be converted into goblet cell, In between, there will be a stage in between goblet cell and stem cell. Stem cell will change it into, is going to change into goblet cell. Goblet cell, we say that it is goblet cell if there is many mucus inside that and becoming like goblet. But if it have a few mucus, it is still columnar but containing less few mucus. Don't accumulate large amount of mucus. It will have oligomucus, so it will be oligomucus cells. Then more accumulation will be mature goblet cell. So oligomucus cell is a transient stage between stem cell and goblet cell due to few amount of mucus. We have in the small intestine is a membrane-like epithelial cell or M cell. It is considered a microfold cell. Let's see how it look like. If you are going to have a simple columnar cell, and this is columnar cell, we are going to have a dome-shaped cell like that. We're going to have a cell like that. It is microfold. It is dome-shaped cell. It will be related to other cells beneath it, which is lymphocyte. So here we have a dome-shaped cell. It is cell like that. It will be related to another lymphocyte, intraepithelial lymphocyte. Can you guess, as it is a cell related to other lymphocyte, what is its function? Yes, this cell will transfer the antigen from the lumen of the intestine into the lymphocyte. So it has a role in the immune system. This cell, as it is related to the lymphocyte, can you memorize where we find many aggregation of lymphocyte? Yes, excellent. It is found in the ileum. Do you remember? In the ileum, there is lymphocytic aggregation or lymphatic follicles, which is called Peyer's patch. So you are going to find this cell many times, or many numerous M cells will be present over the ileum in order to help in the immune response. Let's go. The cell here, by light microscopic section, we can't easily identify this cell because it is very thin and stretched. Can you see here? This is a dome-like cell. It is this cell. It is very thin and can be easily distinguished. This cell do what? Take the antigen from the small intestine to transfer it into intraepithelial lymphocyte, other lymphocyte for stimulation of the. Let's see the electron microscopic picture. As it will be attached to lymphocyte, it will have a number of microvilli in order to help it in its action, so it can increase its number. Now, what about the basal part? The basal part will show many pockets. It will make as if it is like that. This is a pocket. Containing what? Containing intraepithelial lymphocyte and macrophage for transferring of the antigen from the lumen of the small intestine into or to this lymphocyte and macrophage. What about lateral border? Like simple columnar epithelium and goblet cell, it will be attached to other cell by junction complex in order to prevent any passage through intracellular spaces. Cytoplasm here will contain few lysosomes. It is not phagocytic cells. It's just to transfer antigen from the lumen into the intraepithelial lymphocyte. So what to know about lymphocyte? M cell is a cell dome-shaped present in between enterocyte. It is M-shaped like dome-shaped like structure. It is a stretched cell, so can't be seen easily by light microscope. So in order to be seen, it must be by electron microscope. By electron microscope, you will see a concavity inside the cell. This concavity contains immune cells. What are they? They are lymphocyte or macrophage. This cell contains many lysosomes? No, few lymphosomes, because this cell will go phagocyte this cell in order to be transported, not dealing with it, transporting them across the cytoplasm in order to go to antigen presenting cell, which are lymphocyte and macrophage, then going to the mesenteric lymph node to initiate immune response. So is it antigen presenting cell? Is it going to deal with the antigen eating phagocytic and dealing with lysosome. No, it is just transport. So it is antigen transporting cell, not antigen presenting cells. Can you see here? Here we are going to have stem cells. Can you remember stem cells of the stomach? Yes, we was having stem cell in the stomach, in the funding gland. What is the site of the stem cell in the funding gland? It was present in here, in the neck of the funding gland. Here it was found in the neck of the funding gland because it was having isthmus, neck, and base. It is not present in the base, it is present in the neck of the funding gland. What about stem cell in the small intestine? It will be found in the base of the crypt. Okay, so another question. What is the time of complete regeneration of funding gland and small cells of the villus and the crypt? Stem cell in the stomach, funding gland of the stomach, will make complete rapid regeneration in 4 to 7 days. But in the small intestine will make complete regeneration nearly in 3 to 6 days. This cell will find it in many cells will be found in mitosis because it is highly dividable cells. By this, we have finished the first layer of the small intestine, which is submucosa. Now, let's go to the second, is submucosa. What is submucosa? It is dense connective tissue containing blood vessel, nerve, lymphatic, and medullary plexus. But we mentioned that submucosa is very characteristic for small intestine because it will make us identify each part of the small intestine. For example, we say that if we found the Brunner's gland, what is Brunner's gland? They are mucus acini in the submucosa, so this will be duodenum. If was found lymphocytic aggregation lymphatic follicles, this lymphatic follicle is called Peyer's patch. It will be ileum. Excellent. Let's go to see photo and see if we can identify this. Here we can see a photo of finger-like projection. Can you see here? It is finger-like projection. So we are in a small intestine. Here we are going to have a finger-like projection above the surface and deeper invagination here. This is our line. Above is a finger-like projection. This is villus. Below is a crypt. Here is a layer of connective tissue. It is submucosa. What can you see in submucosa? Here, submucosa. We can see rounded structure being in color. This is mucus gland. This mucus gland is called Brunner's gland. This Brunner's gland means that we are in the duodenum. Let's go for more discussing about gland. Brunner's gland or branched tubular gland. What does mean by branched tubular? Means that there is a tube and alveoli. This is branched tubular gland. Branched here. This is branched tubular gland. With this alveoli is considered to be mucus acini, production of mucus. And this will produce their secretion into the crypt. Their duct will open into the base of the crypt in order to pour their secretion into the lumen of the duodenum. So here we are going to have a mucus acini called Brunner's gland. What is Brunner's gland? It is a branched tubular gland opening by their duct into the base of the crypt in order to pour their secretion, their mucus secretion, into the lumen of the duodenum. Why do we need here Brunner's gland mucus? Because here this mucus will be alkaline. What is the importance of alkaline mucus here? This alkaline mucus is important. Why? Because duodenum will receive gastric chyme. What is gastric chyme? It is digested food from the stomach containing HCl. In order to neutralize this HCl, Brunner's gland will secrete alkaline mucus, which will neutralize the acidic chyme that is coming from the pylorus of the stomach, so protecting the duodenal epithelium. It also maintains a pH for pancreatic enzyme because pancreatic enzyme is secreted into the duodenum, so this will maintain the pH for pancreatic enzyme reaction. This was Brunner's gland. So Brunner's gland will be characteristic for the submucosa of the duodenum. Is there any Brunner's gland in duodenum and ileum? No. What about the submucosa of the duodenum? It will be empty. What about the submucosa of the ileum will contain aggregation of lymphocyte. Aggregation of lymphocyte. So aggregation of lymphocyte is called Peyer's patch. Be careful. Peyer's patch or lymphocytic aggregation will be found in the anti-mesenteric border half only. What is mean by that? It means by that if you are going to have a tube covered by peritoneum, this is the tube of the ileum, covered by peritoneum will pass and go to the mesentery. So the anti-mesenteric border here only will be covered, will be containing Peyer's patch. So Peyer's patch is lymphocytic aggregation in the submucosa. What about the villus above this Peyer's patch? Can you see here? Is there any villus above this lymphocytic aggregation? No, it is absent. So submucosa of the ileum containing lymphocytic aggregation. Peyer's patch, it can be easily seen on the ileum. If you take an ileum, you will see a white spots on its surface. This white spots is called Peyer's patch. Is it in the whole circumference of the ileum? No, it is present in anti-mesenteric border. If this is the side of the mesentery attaching the ileum, it will be in the anti-mesenteric border. It is lymphocytic aggregation in the submucosa of the ileum. It is considered one of gut-associated lymphatic tissue. It is one of the GALT. GALT is gut-associated lymphatic tissue. It is having role in the immune defense. So here there will be many lymphocytic aggregation called Peyer's patches. This will reach the lamina propria of the mucosa. Here above the Peyer's patches we will find short or absent villi and many numerous M cell. Is it logic? Yes, because one of the functions of M cell is to transport antigen. And here we're having many lymphocytes, so we need more M cell. So M cell is more numerous in ileum over Peyer's patches, and we need less space for transportation, so there is short or absent villus above this Peyer's patch. Here another picture. Here we are going to have villus lymphocytic aggregation of Peyer's patches above which villus is absent or very short, and this epithelium above it is full of M cell for transportation of the antigen. Can you see here these follicles? Basophilic follicles. This is lymphocytic aggregation or Peyer's patch. Now we can make comparison between three different parts of the small intestine. We have duodenum, we have jejunum, and we have ileum. As regarding duodenum was the shortest. It is about 25 centimeter, taking the C-shaped structure. What about jejunum? It is 2/5. That means it may be 2.5 meter long. The longest is the ileum, which is 3.5 meter long. We mentioned that all of them is going to have a villi and crypt, but the shape of the villus will be different in the three different parts. For example, in the duodenum, you will find that the villus has broad, long leaf-like structure. It is going to have a broad, long leaf-like structure, while the jejunum will be shorter, tongue-shaped. It will be like the tongue, while it will be long, cylindrical, finger-like, least in number over the few in number due to the presence of Peyer's patches. It will be long and cylindrical-like. So when you find that it is a broad, long leaf-like, it will be duodenum. If it is slightly shorter and like the tongue, it will be jejunum. If it is less in number, long, slender, finger-like, it will be the ileum. What about goblet cell? Goblet cells, it will be few in the duodenum, increase in number, and will be more numerous in the ileum. The evident layer is the submucosa, which contain mucus acinoid, contained Brunner's gland in the duodenum. Nothing specific jejunum. If there is lymphocytic aggregation, it will be Peyer's patch. Can you mention other cell different between duodenum, jejunum, and ileum? Yes, it is M cell. M cell will be more evident in the ileum and fewer in duodenum and jejunum. What are the structure and function adaptation of the small intestine? And this part is very important for written and MCQ question. Structure adaptation for digestion. What is the structure adaptation for digestion? Digestion will be started in the stomach, start in the oral cavity. Mostly or the majority of digestion occur in the stomach and fewer occur in the small intestine. This will be helped by secretion of group of glands and enzymes. The first enzyme we present was brush border enzyme, which is called dipeptidase and disaccharidase. Dipeptidase and disaccharidase was found in the glycocalyx of the microvilli. There is secretion of gland from outside. We are going to have two major glands, which is the liver and pancreas. Liver will secrete bile for digestion, pancreatic juice containing digestive enzyme. We are going to have mucosal gland, intestinal crypt, which containing secrete digestive enzyme, hormone, and secretion. Submucosal gland was having Brunner's gland, which secrete mucus in order to protect the epithelium of the duodenum from digestion. What about absorption? About absorption, we are going to have increasing the surface area. How? The intestinal length of the small intestine is about 6 meters. It is very long. Is that enough? No. We are going to have a fold of mucosa and submucosa, permanent fold, which is called plicae circularis. This increases the surface of absorption 3 times more. What about more else? In the mucosa, we are going to have finger-like projection above the surface called villi and deeper invagination into lamina propria called crypt. What else? On the level of the cell to increase its surface area for the function of absorption, we are going to have microvilli that may reach 3000 microvilli per each cell. This will increase the surface area 20-fold. So total increase in the surface area of absorption will be 600-fold. What else? We are going to have secretory function. Does the small intestine make secretion? Yes, it secretes hormones by the enteroendocrine cell. What about immunological reaction? We are going to have and Peyer's patch. Can you see here? Cell number 2. This is called M cell. It is stretchable. It is dome-shaped structure. Can you see here? It is going to have fewer microvilli less than the cell number 1, which is the anterior side. What is the function of this cell, M cell? Taking the antigen from the lumen to go into intraepithelial lymphocyte for immune response and immune reaction. Is it antigen presenting cell? No, it is just antigen transport to transport the antigen into the lymphocyte and macrophage. Now let's have this question. This is a section in the ileum. How we know that this is ileum? Because of the presence of this follicle, which is Peyer's patch. Which part of the ileum is including in this section? How to include this part? Is it mesenteric or anti-mesenteric? Due to presence of lymphatic follicles, it is anti-mesenteric border. Excellent. Anti-mesenteric. How to know that this is ileum? We are going criteria to say that this is ileum. First of all, how we know that this is a small intestine? Due to the presence of finger-like projection here, finger-like projection. You see here, it is a finger-like projection. This finger-like projection means that we are in the small intestine. What else? The presence of deeper invagination, which is the crypt. So this is small intestine. In the small intestine, we have 3 parts: duodenum, jejunum, and ileum. How to differentiate? Look at the submucosa, this region. Submucosa. If it contains mucus acini, Brunner's gland, so it is duodenum. Can you see here any mucus acini? No, so it is not duodenum. Is it empty? If it is empty, containing nothing specific, so it is jejunum. Is it empty here? This is an empty space. Is it empty like that? No, it containing follicles, lymphocytic follicles. So this is ileum. And this is the identifying criteria. No Brunner's gland, presence of the villi and crypt. This is small intestine. No Brunner's gland, no not empty submucosa and the presence of virus patch. This is the criteria for ileum. And here we come to the end of our lecture, and thank you for your time.